In this video, I'm going to introduce you to some of the concepts that lie behind individualized treatment vaccines. And I'm going to start by explaining why it is our immune system should be able to detect cancer cells at all. So the reason cancer cells are visible to our immune system is the same reason why all our cells are visible to our immune system. It's because they're constantly chopping up proteins that they don't need into tiny fragments using those proteasomes. Now, some of those fragments get completely chopped up into individual amino acids, which can then be used to make new proteins, but some of them are left as peptides, small protein fragments, that get processed and then slotted inside little cup structures known as the MHC proteins. And once slotted inside one of these little cups, that protein fragment, that peptide, gets transported to the surface of the cell and gets displayed on the surface of the cell to any passing white blood cells. If a cell is a cancer cell and it contains lots of mutations in its DNA, it's going to be making lots of mutated proteins. Those mutated proteins are going to get recycled, or like the healthy proteins, and hopefully what happens is little protein fragments, peptides from faulty proteins, end up displayed inside those MHC cups on the cell surface for passing white blood cells to spot. If one of these little peptides is from the mutated part of a protein and it's weird and abnormal and likely to be spotted by the immune system, we call it a neoantigen. Some cancer cells display a lot of neoantigens on their surface. For example, if they are a cancer that has developed because of the effect of UV light from the sun, a skin cancer perhaps, or if it's a lung cancer that's developed because of the DNA damaging effects of cigarette smoke, it's likely to have lots of neoantigens on its surface. But if a cancer cell is driven by a handful of powerful mutations rather than the accumulation of thousands of small mutations, if it contains fewer mutations, it's less likely to have neoantigens displayed on its surface. And cells with fewer neoantigens are likely to go undetected by our immune system. Treatment vaccines, the goal of them is to provide neoantigens to the immune system, to teach the immune system what neoantigens might be present on the surface of the person's cancer cells. And I'm going to pick out one of these personalized, individualized mRNA vaccine strategies as an example. Now, this is a complicated diagram, so I'm going to go through it one little bit at a time. We will start with cancer cells taken from a cancer patient. These cancer cells are compared and contrasted to the DNA extracted from white blood cells from that same patient. The white blood cells are being used as a source of healthy DNA to be compared to the cancer cell DNA in their body. And scientists try to pinpoint mutations present in all the cancer cells in the patient's body, not just in subsets. Having identified these really important mutations, the scientists then try to predict which of these mutations lead the cell to create faulty proteins from which neoantigens might be derived that are then going to be in the MHC cups on the cancer cell surface. So they're working backwards from the DNA and trying to predict what the neoantigens as a result of those mutations might look like. And then they go back a step more, but from that prediction of what the neoantigens are, they try to predict what the DNA, the gene, would look like in order to lead to that neoantigen. So they predict what the mutations are, what the mRNA would look like that the cell is then using to make the neoantigens. So they then design, in this example at least, they design a single strand of mRNA containing the instructions for up to 34 of the neoantigens they've predicted to be possibly displayed on that person's cancer cells. mRNA is very fragile, so they have to protect it in some way, and they do that by creating tiny little lipid droplets that contain that precious mRNA cargo. You then inject the mRNA into muscle, passing patrolmen of the immune system, dendritic cells. They pick up those little lipid particles and the mRNA they contain, and they use the mRNA to make the neoantigens. They then display those neoantigens inside their MHC cups. They go off to nearby lymph nodes and they start talking to T cells and activating T cells. Hopefully, the dendritic cells create lots and lots of activated T cells, which now go out into the body and seek out and destroy cancer cells with those neoantigens on their surface. So you've started off with what mutations exist in the cancer cells. From that, you've predicted what faulty proteins they're making. From that, you've predicted what neoantigens might be on their surface. And then you've tried to introduce those neoantigens into the body. And in this example, they've put in the mRNA for those antigens. The dendritic cells pick up the mRNA, they make the neoantigens, they show them to T cells. The T cells become activated, and hopefully the T cells seek out and destroy the person's cancer cells. So far, we've seen some preliminary results from clinical trials with this kind of approach. This is one example of one such trial using the method that I've just tried to explain. And in this trial, patients were randomized to receive either their personalized mRNA vaccine plus pembrolizumab, an immune checkpoint inhibitor, or pembrolizumab alone. And they were then monitored to see, after surgery, having received this treatment, which patient's disease returned, which patient's disease did not return. And the patients who, following their surgery, had received their personalised vaccine and a checkpoint inhibitor, they were less likely to see their cancer return than patients just given the immune checkpoint inhibitor. The vaccine did take between 7 and 9 weeks to create for each patient. While they waited, they received just the immune checkpoint inhibitor. Because of the results of this trial in melanoma patients, there are now 4 phase 3 trials underway, 2 of them involving patients with non-small cell lung cancer, 1 involving patients with squamous cell carcinoma, and 1 with patients with melanoma. A little summary of those vaccine treatments. There are lots of these sorts of treatments in development. I've just shown you one strategy where the mRNA for neoantigens is introduced into the body, but you could try and introduce just the neoantigens themselves. You can even take dendritic cells out the body, feed them those neoantigens and put them back into the body again to recruit and activate T cells. There's lots of different ways being developed as to ways of wakening up to the immune system and teaching it what cancer cells look like. The difficult part of this is generating a strong enough T cell response to have a meaningful effect on the person's cancer cells. There have been many clinical trials in which treatment vaccines have failed to make a difference, despite you being able to prove, yes, there's lots of T cells, yes, they look great, but there's just not enough. They're not able to overcome the person's cancer. Ideally, you need to target these vaccines at multiple different neoantigens, not just one. You need to give them to patients who have as little disease in their body as possible in order to give the vaccine and give the T cells a fighting chance to overcome and overwhelm those cancer cells. And you have to add in powerful adjuvants, things that boost the immune system and give the alarm signal to the immune system.